is very important for the public to know that cancer prevention is possible.
Each year, around the world, roughly 20 million people are diagnosed with cancer, and close to 10 million die from it. About 1 in 5 of us will face it in our lifetime. For decades, the fight against cancer has followed a single pattern. We wait. We watch for symptoms. We screen. And once cancer appears, we treat it with surgery, radiation, chemotherapy, and a generation of newer therapies that have saved millions of lives. Treatment has improved dramatically, but it begins only after the disease itself has already taken hold. Tomusiak, founder of Permanence Bio, is determined to break that pattern and put us on a different trajectory entirely, one that intervenes long before a tumor ever forms.
The analogy that I like to use is, imagine if our airline industry, to maintain safer flights, focused entirely on how to keep a plane upright once it's already falling from the sky. It would be incredibly inefficient to be targeting airline safety in that way. What I'm working on and what the field is gradually moving towards is, okay, like, how do we keep the airplane in the sky in the first place? How do we prevent that collapse from happening?
His strategy works at the root of the sequence, shielding our DNA from damage so the mutations behind cancer never form.
We want to prevent the core cause of cancer, which is mutations. If you do not ever get DNA damage, you don't get mutations, and mutations are necessary for cancer formation. We're developing small molecule drugs that shield DNA, prevent it from getting damaged in the first place. If there is DNA damage, these small molecules will help in repairing the DNA correctly so that cancer never actually happens.
Unlike antioxidants, which scatter through the cell and hope to be nearby when damage hits, Permanence Bio's molecules lock onto the DNA itself, right where cancer begins. For the young founder, the urgency is personal.
My father was sick with cancer, and he was diagnosed with a melanoma that had ended up spreading to his brain. He was originally given 6 months to live, and then about a year and a half later, he was still alive. But the side effects of the treatment were brutal. He had gone from radiotherapy to immunotherapy to steroids to radiotherapy to steroids to, back all over again. And I couldn't shake this feeling that it could have been better had we prevented the cancer from initiating in the first place, or if it was possible to at least prevent it from spreading, that acting earlier in the cancer treatment process and instead of way at the end.
A decade ago, an idea like this would have drawn skepticism. Today, with serious companies racing at the moonshot edge of DNA repair, Permanence Bio represents the frontier of disease prevention. And the startup is already seeing promising results.
We looked through all the literature on every, antioxidant, the vitamins, the vitamin C, various different types of compounds that have been previously shown to prevent DNA damage. And the most promising lead came from a pretty surprising source. It was from a late 70s U.S. government program to develop radioprotectors. These were compounds that would be administered to U.S. warfighters and civilians in the event of a nuclear attack by the Soviet Union. These studies were massive. This was like a U.S. military study where there was a base and there were 7,000 different compounds that were tested to screen at scale the best possible DNA protecting compounds. And they found a few that worked remarkably well. So what we do is we take, a library of pre-existing compounds. We then take the best existing U.S. military radioprotectors. At this point, we've created like hundreds of derivatives of them that we can get IP around, and also we have reason to believe will work better. And then we screen them in cells. So we look at, normal cells, healthy cells. We hit them with radiation. We hit them with low-dose radiation, high-dose radiation, formaldehyde, like UV damage, any sort of damage that you could imagine a human experiencing in their lifetime. We try and hit these cells with these compounds, and then show that these compounds can actually protect against that type of damage. We then also do the same thing for cancer cells. So we don't want to protect cancer cells. If you protect from DNA damage, you might be doing more harm than good if you protect cancer cells. So we have a counter screen where we take the same, batch of compounds, the same types of damage, and we screen them against cancer cells to make sure that the cancer cells are not protected. From there, we basically get a pretty massive database of data. We look at, not only do these cells survive, but also what do they look like all the way from, all the cells alive or dead, to what do they look like under a microscope, to all the way to like RNA sequencing. Do the cells look healthier? Do they look happier? Or are they like upregulating pathways that are not good for humans? Ultimately, the vision is to take that database and then fuel it with AI to then create novel compounds that are going to be even better than the ones we just tested. But then we take the best hits from that database and we move them into animal models. Basically, there's animal models of cancer initiation where these animals get cancer on a relatively early basis, and then our compounds are being screened there to show, hey, like for these animals, can we prevent their cancer from progressing in the same way they would prevent a human cancer from progressing? We have a few compounds that work really well in animals. We've shown cancer prevention and tumor initiation drop by about 50%. We're hoping to initiate clinical trials in the next 2 or 3 years.
The fight against cancer is now also a fight to prevent it. And in that change lies the most hopeful possibility in modern medicine, that a world without cancer is not a question of if, but of when.